thank you. It's a privilege to be here and tell you about OncoSpherix and our work on developing small molecules that disable hypoxia-driven gene expression in cancer. OncoSpherix is a new company. It was founded in 2018 based on work out of Emory Universities and Georgia State Universities. Erwin Van Meer is one of our scientific founders. He was at Emory at the time and has now relocated to the University of Alabama at Birmingham. And then Bing Wang is our other founding scientist who's at Georgia State University. Our intellectual property, we've licensed technology from Emory and Georgia State Universities. And specifically, we have first-in-class dual HIF-1 and HIF-2 inhibitors, as well as a family that inhibits both HIF family members and mitochondrial complex 1. We have composition of matter and use patents, US and international, and additional IP filings are planned. So we are a new company. The licensing was completed roughly a year ago. I'll be telling you about the importance of our targets, HIF-1 and HIF-2, the screening strategy that was used, efficacy in multiple mouse models of human cancer, the mechanism of action, the advantages of our drugs compared to other agents that have effects on HIF, how we've been able to combine our lead candidate with cytotoxic chemotherapy, and our plans for advancing the lead candidate towards clinical testing. So let me begin by telling you a little bit about hypoxia in cancer. It's a very common feature because many tumors grow faster than their blood supplies. And the hypoxic areas are alive. There are many tumor cells in the hypoxic areas that are alive, and they play a major role in the spread of cancer to new areas. Now, in this slide, I'm showing you a malignant brain tumor. This is an experimental mouse model system, but the brown area, this was stained with the oxygen-sensitive reagent pimodidazole. And this shows the hypoxic region, whereas there's a blood vessel here and here. And you can see that you don't have to get very far from the blood vessel before hypoxia starts developing. It's about 10 to 15 cell layers. And the cells that are in these hypoxic regions, particularly in the initial areas of hypoxia, are very biologically active. They express HIF-1 and or HIF-2 as master regulatory proteins to drive expression of other genes that help the tumor cells survive and spread from these regions of hypoxia. Both HIF-1 and HIF-2 are expressed in multiple types of cancer, primarily in the hypoxic region. And at one point it was thought that there would some tumors that would express primarily HIF-1 and others that would, a much more limited subset expressing HIF-2. But it turns out that both HIFs are expressed in a variety of different types of cancer. And I'm showing you an example here that's a resected, actually two different surgical samples, resected glioblastoma multiforme. And here's the blood vessel here. And as one gets, again, about 10 to 15 cells from the blood vessel, you start seeing the HIF that's stained brown. And this is HIF-1 alpha, which is the oxygen-sensitive subunit of HIF-1. And this is HIF-2 alpha, the oxygen-sensitive subunit of HIF-2. So both can be expressed in a number of different types of cancer in response to oxygen starvation. Now, this is a simplified schematic of how oxygen affects the alpha subunits of HIF, which are the oxygen-sensitive forms. And under normoxia, the alpha subunits become hydroxylated, and then this targets them for degradation by the proteasomal degradation system. Whereas in hypoxia, they do not get hydroxylated, and they are able to recruit the P300 CBP subunit, cofactor, as well as alpha-1b subunit, form a transcriptional complex. So the stabilization of the HIF alpha subunits allows the formation of the transcription complex and subsequent transcription. So our goal was to develop a drug that blocks HIF-driven processes. And so just to recapitulate, as solid tumors grow, there are regions of hypoxia within the tumor. And as a consequence, HIF-1 or HIF-2, the alpha subunits become stabilized. That leads to formation of a transcription complex. And when you have that active complex, it drives a number of processes through the genes that are driven, such as angiogenesis with VEGF receptor being expressed, a number of growth and survival signals, including MYC and cyclins, also multi-drug resistance genes contributing to the resistance to standard chemotherapy. Genes involved in invasion and metastasis, including CXCR4, c-MET, cathepsins, and things that alter glucose metabolism, because in the hypoxic regions, then there's a shift to glycolysis as the major energy source with a loss of oxidative phosphorylation. So our goal was to develop a drug that would block the ability of HIFs to drive all of these processes. And our strategy was to use a reporter system, a HIF-driven reporter system. More than 10,000 small molecules were screened and hits were identified. From those hits, structure activity studies were done to improve potency. There were about 500 tested. From one was selected for initial efficacy and mechanism of action testing. From that, then additional chemical modifications were made to improve potency and solubility. And ultimately, we selected a lead clinical candidate that we call 64B. Not a very glamorous name, but that's what we're currently using. So in addition, we do have some backup compounds of the same class that have some distinct medicinal properties, and then some follow-up compounds are in development. So I'd like to show you that 64B is effective in a variety of different types of cancer, in both affecting primary cancer growth and metastasis, as well as prolonging survival. And so in mouse xenograft studies for triple negative breast cancer, adenocarcinoma of the lung, or ocular melanoma, allowing tumors to establish and then treating them with 64B, there was a dramatic reduction in tumor growth in all three types of cancer examined. So this is primary tumor growth. And when we took one of these, actually we've looked at all of them. I'm just showing you one here, which is in lung cancer, looking at the metastasis. Lung cancer metastasizes to the contralateral lung in this particular model, and looking microscopically for evidence of metastasis. In the controls, you can see tumor nodule here, which when stained with Ki-67, which identifies highly proliferating cells here, we can identify the nodules and count them in the lung. And when looking at 64B treated mice, no nodules are identified. And so in this particular case, it was done in 10 mice. You can see the span of metastatic nodules per mouse in the control and 64B treated. So we're showing that we can block both primary tumor growth as well as inhibit metastasis with using 64B as a single agent. This is another model looking at ocular melanoma. Now, ocular melanoma has distinctions in its pathogenesis and response to therapy compared to cutaneous melanoma. This model, the tumors are implanted in the eye, so one can look at primary tumor growth. And then starting treatment the following day, remove the eye after a week, and then let the mice continue to live for 28 days, then sacrifice them and look for metastasis in the liver. And two different models. One is syngeneic, the other is a xenotropic. It's in nude mice, so human tumors in nude mice. And looking at the primary tumor here versus treated, here versus treated, there's about a 70 to 90% reduction in primary tumor growth. Then taking the livers and counting the number of metastasis, then there's a dramatic reduction in metastasis in both the syngeneic model and then in the xenographic model. When we allow the mice to, similar experimental structure, but allow the mice to go using survival as an endpoint, we also see prolongation in survival with 64B. Now that's using 64B as a single agent, but in fact, 64B is not cytotoxic, and it's really designed to work primarily in these hypoxic regions, and so it's really designed to be given with other agents. So we asked two questions. Can it be effectively combined with cytotoxic agent? And in this case, it's doxorubicin, doxycycline. And is there evidence of synergy? And then second, how does it affect the toxicity? 64B is minimally toxic, whereas doxycycline is a cytotoxic agent with significant toxicity. And as a single agent, the 64B is as effective as the doxycycline in inhibiting primary tumor growth. But when we put the two together, you see a dramatic reduction in tumor growth over time. And so this is looking at the average of 10 mice. This is at the final time point when the tumors were harvested, weighed, and each dot represents a separate tumor with the mean and the p-values shown here. So how does it work? Well, we have shown that, okay, so first, when this ends up showing that during normoxia, which is 21% oxygen, HIF, the alpha subunit, in this case HIF-1 alpha, is not expressed. Oxygen starvation induces expression of HIF-1. This is done in tissue culture. And 64B does not block that. And so this is shown in, let's see. Okay, so here we're looking at CBP, and here we're looking Western blot at HIF-1 beta. So this is the input. This is immunoprecipitation using antibodies to HIF-1 alpha. And you can see that the CBP is interacts with the HIF-1 alpha in the absence of 64B, but not in the presence. And it has no impact on the interaction of HIF-1 beta. Thermal shift assays also show that 64B, which is shown right here, binds to P300. So the structural elements that are required for the binding of P300 to HIF-1 and 2 are conserved and are found on the CH1 domain of P300. Now this is a linear representation of CBP300. While these are two separate proteins, distinct proteins, they're structurally so similar that they're often grouped together. And in fact, HIF-1 alpha and HIF-2 alpha bind to both. It's on the CH1 domain, and this was work that was done back in 2002 by Friedman et al. And while there are a number of other proteins that interact with this specific domain, importantly, the acetylation domain, histone acetylation domain of CBP300 is not affected by the HIF binding site, is not involved in the HIF binding site. And there are a number of other binding sites for a number of other important transcription factors that are distinctly located. Now, when you look at a linear representation, it doesn't take into account the folding, and so the structural elements of the interaction between HIF-1 alpha, HIF-2 alpha, and CBP300 have been defined, which is very helpful when we go and try and understand how 64B interferes with that interaction. So some modeling was done, and for full disclosure, it was done with an initial molecule that we were using that we have shown is functionally identical to 64B, called KCN1. And so here it shows the binding of it to the CH1 domain of CBP here and here. And so, important things to keep in mind is that these drugs are not general CBP300s. The binding sites for these are, for CBP300 for many transcription factors are not affected, and they do not bind to the histone acetyltransferase site. We've shown functionally that P353 transcription is not inhibited, and toxicity from 64B is minimal, which is not the case when one has a general CBP-P300 inhibitor or one that affects the histone acetylase transferase site. So other groups have developed some drugs that either directly or indirectly affect HIF expression or function. Some of them will decrease the signals that lead to HIF mRNA expression. The two most specific that have been tested, one is an antisense to HIF-1 alpha that has no effect on HIF-2 alpha. And then one is something that affects the dimerization between HIF-2 alpha and HIF-1 beta. This is something that is in clinical testing right now for renal cell carcinoma and has shown some efficacy. So with our drug, it actually affects the recruitment of P300 to the transcriptional complex. And so it blocks the function of the HIF complex without affecting the levels of either HIF-1 alpha or HIF-1 beta, which I just showed you. There's some other drugs that can affect transcription, but they're very nonspecific to HIF. So ours is the first dual HIF-1, HIF-2 inhibitor, and the mechanism of action has been defined. Our preliminary toxicity studies in tumor-bearing mice show no overt signs of toxicity when given at therapeutic doses, both alone and in combination with two different cytotoxic agents. No evidence of injury to major organs when examined histologically and serologically at the completion of daily treatment. And our IND-enabling toxicity studies will be done using a clinical formulation that we have yet to develop. So in summary, 64B is a small molecule inhibitor of HIF-1 and HIF-2 transcription factors. It blocks the survival and spread of cancer cells that are triggered by hypoxia. It blocks neoangiogenesis, including blocking genes such as VEGF and VEGFR, things that are blocked by and some commercially available angiogenesis inhibitors, but it does so more thoroughly through blocking the expression of a variety of different genes. It blocks invasion and metastasis, and it blocks the ability of cancer cells to survive in a hypoxic environment. 64B is not targeted therapy, except in rare instances where tumor cells are HIF dependent, such as in renal cell carcinoma that have deletions or mutations of the von Hippel-Lindau protein. It is not immunotherapy, but does affect the immune response to treatments through altered gene expression. It's not directly cytotoxic, and it's not an inhibitor of hypoxia. Hypoxia persists, but it prevents the responses to hypoxia. So these properties allow 64B to be effectively combined with other treatments, increasing cancer control in a relatively non-toxic manner. So we've identified our lead candidate. We have in vivo efficacy data in multiple types of cancer in mice. We've shown some therapeutic synergy, which we want to expand on. We've defined the mechanism of action. We have toxicity in tumor-bearing mice, and we have some non-dilutive funding. Our next steps are to optimize the pharmaceutical properties of 64B through both pre-formulation and formulation studies. We need to test additional combination therapies for efficacy in preclinical models. And once we have our formulation complete, then execute IND-enabling toxicity studies. Our current plans on clinical trial priorities using 64B in combination with another agent is for relapsed glioblastoma multiforme, high-risk ocular melanoma, and unresectable adenocarcinoma of the lung. So I will stop there. Thank you for your attention, and I'd be happy to answer any questions.
Thank you, Dr. Huffman. That was an excellent presentation, and you generated a lot of interest from the audience. So I'll begin with the first question is, are normal angiogenesis, growth and survival, and glucose metabolism under regulatory control by a different regulator other than HIF?
So HIF ends up playing a major role in that. So with knockout studies, then both HIF, you have redundancy between HIF-1 and HIF-2. So when you end up knocking out one, you can end up getting some angiogenesis or some changes in glucose metabolism from the other. Where one of our scientific founders, Erwin van Meer, is creating some knockouts right now to specifically be able to more completely answer that question. So yes, there are other factors in addition to HIF that contribute to it, but we do see with our drug, when we disrupt HIF-1 and HIF-2, a major disruption of angiogenesis and tumor survival.
Okay, thank you. Another question is, do you have any effect on the primary tumor in the eye, or does 64B only target metastasis in secondary organs?
So we do have, I showed you, and I know I ran through things pretty quickly, but in that mouse model that I showed you, the tumors were implanted on day 0. On day 1, 64B was started, and on day 7, the eyes were removed and then looked at the tumor. And so there was a 70 to 90% reduction in the primary tumor growth or size in response to 64B in the eye. In addition, there was the reduction in the metastasis. Now, we've just gotten some SBIR funding to expand those studies into a rabbit model, which allows us to actually monitor in more real time what's happening in the eye. And we'll also be combining 64B with some other agents to more completely define the impact in the uveal melanoma.
Okay. Next question is, do tumors of the liver exhibit hypoxia? And if so, any more or less than other tumor types?
So tumors of the liver do display hypoxia. And I know there have been some studies that have been done defining how hypoxic different tumors are compared to to normal tissue, measuring the actual oxygen tension. And I confess, I don't remember where hepatocellular carcinoma fits on the spectrum compared to some other tumors, but that information is available.
Okay, thank you. Another question is, how do you administer 64B into mice? And are there any other adverse effects such as liver toxicity?
Okay, so the way we have been administering it for these studies has been through intraperitoneal injection, which is clearly something we would not be doing in humans. And so that's where now that we have some good efficacy data and some good evidence of stability and other pharmacologic properties. A major focus will be on formulation issues to determine whether we can administer this orally or whether we need parenteral administration. So I'm talking to a number of groups right now to basically explore our options, but that is a very high priority for us to be able to switch from IP to hopefully oral, and with our rabbit studies, we want to complete our formulation work before doing the rabbit studies.
Okay. And then one final question. Great talk. What do you think of HIF in other indications like PAH?
PAH.
I'm assuming that's pulmonary arterial hypertension, maybe?
Okay. Okay, just making sure I'm understanding the question. I am not familiar with the literature on that, so I won't venture a guess.
Okay, fair enough. We did get one more. How is the efficacy of 64B compared to HIF-2A inhibitor like PT2977? It's a MK6482. I'm assuming it's a Merck drug.
Right. So that is a drug that is currently being tested against renal cell carcinoma. It's specific for HIF-2, whereas our drug is a dual HIF-1, HIF-2 inhibitor. And that drug the HIF-2 inhibitor has shown evidence of efficacy as a single agent with the VHL mutant renal cell carcinoma. We anticipate our drug will be as effective for that indication, but we also anticipate our drug will be superior for a number of other indications because it inhibits both HIF-1 and HIF-2. And the number of tumors that are exclusively dependent on HIF-2 are few and far between. So we haven't directly compared it yet, but we do plan to.
Okay. These questions will be available for you, Dr. Offermann. We can get those to you through SAM. So if you want to have any specific follow-up with the individuals that have asked the questions. And we thank you very much for an excellent presentation. Thank you.
Thank you very much.
There is one last question, sorry, from Brazil actually, and he's asking, what is the brain exposure of the compound?
Good question. Our original compound, which was called KCN1, did not have good blood-brain penetration, and so 64B, we're seeing at least 70% of the serum levels in the brain within an hour, but we do need to do more detailed studies. And so part of our interest in developing 64B is because of its ability to penetrate the blood-brain barrier. So that is work that is currently in progress right now.
Excellent. Okay, thank you very much again.
Thank you.